Good morning, everyone. I'm so happy to moderate today monthly webinar from the International Society of Bone Morphometry. Um, Actually, I would like to start with a couple slides talking a little bit about the society, and then we will present our speaker today. So the ISBM, or the International Society of Bone Morphometry, it's a society that's interested in studying all the quantitative assessment of bone across scales. And actually, the nice thing about the society is that we have really wide varieties of opportunities for leadership, for established investigators and early career investigators as well. In addition, in the society, there are different aspects of research interest. Like, for example, we have different working groups for whoever is interested in advancing protocols for studying bone in clinical research. In addition, we have also another one that's interested about spatial transcriptomics in skeletal tissues, imaging and analysis of nerves and bone microstructures. And I would say this is a great opportunity for people to network and meet and collaborate. So we're really excited to invite all of you who are not members to join us in the ISBM. So we happy to announce that we're gonna have the annual meeting of the ISBM 2026 is gonna happen at July 5th and 6th in England, and still the registration is open. So please, we would be happy to have you and attend this meeting. We have a lot of keynote speakers, and really there is a wide range of opportunities to have research talks and clinical talks. So please, if you have the chance to come in England, and we will be happy to see all of you to attend this meeting. In addition, if you would like to follow our news and updates, and in addition, the announcement of the future talks, please follow us on the LinkedIn page. We don't usually bother people with a lot of posts and emails, but it's really good way to follow up our informations. So today monthly webinar is kind of organized by the early career investigators of the ISBM. And please, if you are an early career investigator and interested to join us, don't hesitate and shoot us an email, and we will be happy to host you in our society. We would like to thank Alexian for supporting this series of webinars, which makes really us happy to share with you all the information we need or we know about bone morphometry. I think I have been talking, but I'm so happy to present our speaker, Dr. David Ruffoni. And I'm not good in Italian, Dr. Ruffoni, so I hope that I spelled it in a good way. And today's lecture is titled Distinctive Features of the Cement Line in Human Osteonal Bone. And Dr. David really has a really rich resume. So he had his PhD from Max Planck Institute in colloid and interfaces, and he really had a very rich postdoc experience in Zurich. He had really a diverse research interest between implant biomechanics and bone biology with Dr. Ralph Muller. So I'm really excited and waiting to hear your presentation, Dr. Raffoni. And sorry for the long introduction, but now you have the stage for your presentation. Welcome.
Yeah, thank you very much for the kind introduction and also for inviting me to give this webinar. I'm going to share my presentation. So, do you see it?
It's loading. Yeah, working now.
Okay, perfect. Yes, so basically, yes, I like to present a little bit of our research that we have been doing on the cement line. And as introduction, just I look for the pointer, 1 second. Do you see also the arrow of the mouse?
Yes.
If I move, yes, great. Yeah, just as introduction, I like to, well, remind you that bone is a beautiful hierarchical structure let's say, spanning many length scale from the nanometer up to the millimeter and even centimeter length scale. This is something that we all know. A little bit maybe less known is that the concept of having a hierarchical structure means that many building blocks are connecting together at different hierarchical levels, and these building blocks are connected through numerous internal interfaces. And one of those interfaces is the cement line, and this is what we are interested in. Specifically, let me switch. Voila. The laser is here. Yeah. Specifically, the cement line is a very tiny interface, and we can find this in many places in bone. We can find this in cortical bone around secondary osteons. We can find the cement line in trabecular bone joining different bone packets together. And also we find this at the interface between, let's say, bone and soft tissues at the osteochondral junction. The cement line joins mineralized cartilage to subchondral bone. And at enthesis, the attachment of tendons into bone, the cement line is also there to join mineralized fibrocartilage to bone. And also to some extent at the bone implant contact, there is a first layer which is very similar to the cement line. Why are we interested in the cement line? Actually, well, the cement line provides a substrate for bone deposition, is actually in the deposition of the lamellar bone, is the first layer on which the osteoblasts are, let's say, sitting and depositing lamellar bone or osteoid that then is mineralized into lamellar bone. From a biomechanics perspective, there are both experiments and simulations which have shown that, let's say, if we consider cortical bone, cracks, depending on their length and trajectory, tends to, let's say, deviate and avoid and, let's say, deviate around osteons. So they are running around the osteons and they do not enter in the osteons. And so cement line has been assumed to have the ability to interact with damage and can be, for example, for instance, considered one additional aspect of bone quality. And this is more for a biomechanics point of view. And last but not least, from a mechanobiology viewpoint, we all know that in bone, the bone is populated by cells, osteocytes, which form this interconnected network, the osteocyte lacuno-canalicular network. This network is very important for, let's say, transport of matter, of information, for the mechanosensory ability of bone. And an interesting question for us is to understand if let's say, the interaction between the osteocyte lacuno-canalicular network and the cement line, and the ability of the network to cross the cement line and to allow communication, exchange across different osteons or between osteon and interstitial bone. So these are a little bit the aspects that I would like to, let's say, discuss today. Some features which are known about the cement lines, which I would like to remind to you. Well, we call it cement line, but in reality, the cement line is a 3D structure. The 3D structure of the cement line is experimentally accessible, for example, using synchrotron radiation, nano-CT, or a FIB-SEM. One other thing that is now well accepted is that the cement line has a higher mineral content than the surrounding tissues. And the cement line is also rich in osteopontin. Osteopontin is a protein which has high affinity to calcium and is considered an interfacial adhesion promoter. So I'm not entering, in my talk, I will not discuss osteopontin, but then I refer to you to the work of Mark McKee, for example, on this topic. I like also to remind to you, or to remind us, let's say, how we, let's say, the most common picture on the formation of the cement line in osteon. So the basic idea is that we have the classical cutting cone that is forming secondary osteon, where the osteoclasts are removing bone. And then this is followed by, let's say, pre-osteoblasts that are acting together with what we call secondary osteoclast. They combine with secondary osteoclast, and they are cleaning a little bit the bone surface and preparing the bone surface for subsequent bone deposition. And is this actually are those type of cells which are probably in charge of depositing the cement line. this is also nicely investigated in a work of Lassen and colleagues, where they investigate actually the spatiotemporal organization of BMUs, and even they came up with a very nice 3D reconstruction of how this process is working, and is actually during the reversal and resorption phase by the where there is, as I said before, the combining presence of let's say secondary osteoclast and pre-osteoblast. And in this region is in this region where the cement line is formed before then there is before the formation of lamellar bone by osteoblast. So this is a little bit the warming up part of my presentation. And now I'd like to enter a little bit more into our research question that we have. In principle, we are interested to understand a little bit more the material properties of the cement line. I said that the cement line is now, there is consensus that is more mineralized than surrounding tissue, but what we still do not know too well is the mineralization process of the cement line. So if we depict here a newly formed osteon surrounded by the cement line and the interstitial bone outside here, what we are interested to understand is how the mineral content of the osteon and of the cement line evolves with time. So we are interested to understand the mineralization process of the cement line. We are also interested to understand the mineral properties of the cement line, so that really the mineral particles, the thickness, the length, their orientation, and how is there, if we ask ourselves if they are different from the properties, mineral particles of surrounding tissue. And this is something which may be relevant for bone quality. We are then also interested in the mechanical properties of the cement line. And this is, of course, relevant for bone fracture resistance. And particularly, we are interested to understand if the cement line is stiffer or softer than surrounding tissue, and also the composition mechanics relationship of this interface. And last aspect, we are more, let's say, focusing on the biological properties and on the interaction between the cement line and the lacuno-canalicular network, which may have implication for the transport of, as I said before, transport of matter and information, let's say, between different osteons, and also for the mechanosensitivity of cortical bone in this case. So this is basically the PhD project of my former PhD student, Astrid Cantamessa. And this is something that we actually, let's say, look at those questions following a multimodal correlative analysis with also colleagues. This is a collaborative work with colleagues in the Ludwig Boltzmann Institute for Osteology in Vienna, colleagues at Max Planck Institute of Colloids and Interfaces in Potsdam, in Germany, and also with the, let's say, we performed experiment at the European Synchrotron Radiation Facility in Grenoble. So the material for analyzing, let's say, the mineralization of the cement line, we used quantitative backscattering electron imaging, which is a technique allowing to have two-dimensional maps of the mineral content of the cement, or let's say of the tissue that we analyze. Mechanical properties are basically investigated with nanoindentation, so which has approximately the same spatial resolution of QBI. And mineral properties are investigated with a wide and small angle X-ray scattering with synchrotron radiation, and the biological properties with the confocal laser scanning microscopy and on samples which have been stained with rhodamine, which access the accessible porosity. And what I like to underline here is that the small thickness of the cement line, which is only into dissection of about, let's say, from 2 to 3 micrometer, requires investigation methods which have sub-micrometer resolution. And we apply all these methods in a correlative manner, so meaning that we have analyzed the same samples, the same locations with the same resolution. And then we combine all the information together and to try to extract a little bit, let's say, new aspects of this interface. So maybe I start to present, I'm presenting basically summarizing here 3 main papers that we had on this. 2 are already published and 1 is under submission. And I'm presenting now the first paper that we did on the mineralization of the cement line. So in this context, there is a previous work of Milovanovic and colleagues who have analyzed already, let's say, the cement line in osteonal bone and have investigated the mineral content of the cement line with respect to the mineral content of, let's say, the corresponding osteon. They did this analysis considering several osteons. And if we, let's say, the main finding is the following. If we plot here the mean mineral content of the osteon on this horizontal axis and the mean mineral content of the cement line on this vertical axis, we see that the cement line is always more mineralized than the osteon, and the difference between the mineral content of the cement line and of the osteon decreases as the mineral content of the osteon increases. I'll come back to this later on also when I discuss my result, but if we interpret here the mean mineral content of the osteon as a, let's say, as a proxy for tissue age, the older are the osteon, the more similar is the mineral content of the corresponding cement line. So this is a little bit one starting point also to understand our, let's say, our work that we did. Before presenting our work, I also like to mention that a recent work by Tang and colleagues from the group of Peter Fratzel analyzed also the cement line with FIBSEM and also came up with an explanation for the higher mineral content of the cement line. Because what they found is that there is, in addition to the, let's say, canalicular network, in bone, there are also nanochannel, or maybe better nanopores, which have dimension of about, let's say, less than 30 nanometers in diameter. So they are much smaller than the canaliculi. And these nanopores are present in bone, but are not present too much at the cement line. So if we look at the cement line here is a region in the osteon which has two cement lines which are close by. Those cement lines have a peak in mineral content and have a minimum in the, let's say, volume fraction of nanopores. So one possible explanation for this higher mineral content, which is measured with the QBI, is that in the volume which is assessed by QBI, which is about 1 cubic micrometer, well, in this volume, there are much less nanopores in the cement line than in the adjacent tissue, and this contributes to a higher mineral content that we see. Okay, so this is a recent work that I wanted to mention. But now coming back to our research question, well, let me remind you what we know a little bit about the mineralization process. As you know, when osteoid is deposited, osteoid mineralizes very fast. In a few days, it reaches maybe up to 80% of the total mineral content that can be accommodated inside the collagen matrix. And then over several weeks, up to months, if not years, there is a very slow secondary mineralization. In this fast primary mineralization, the mineral particles mainly increases in length, and in the slow secondary mineralization, the mineral particle increases in thickness. And what we are interested in understanding is if we can, let's say, extract a little bit, we can extract the mineralization process of the cement line in comparison with the mineralization process of the corresponding osteon. So this is a little bit our research question. So to do this, we analyze human femoral bone from two individuals, and we focused on uninterrupted osteons. So these are the osteons which are the most recently deposited in the, let's say, in the location that we have analyzed. And we performed a layer analysis similar to the one proposed by Milovanovic, but we do not only considering the cement line, but also considering bone located very close to the cement line, both in the osteon and in the space outside the osteon. And the space outside the osteon can be composed of other osteon or interstitial bone. So we subdivided our region of interest in several layers, in the cement line and in layer very close to the cement line inside and outside the osteon. So this is now a little bit a magnified view. The cement line is here in orange, a layer very close inside. They have a distance of only, let's say, 1 micrometer from the cement line. up to 10 micrometer away, and also inside the osteon and outside the osteon. And then we look at the mineral content inside those layers, and we look for correlation between the mineral content of the cement line, the mineral content of layer very close to the cement line. So this means that they were deposited soon after the cement line inside the osteon, and also very close to the cement line, but outside the osteon. And so this means that they were deposited much before the cement line was formed. So that's a little bit the concept that we are considering. We also use second harmonic generation imaging to make sure that the selected osteon were uninterrupted, and so the most recently deposited. So once we measured all these osteon with QPI, so we could extract the gray level here is proportional to the local calcium content. And then we, let's say, analyze the mineral content in the layer, for example, considering this frequency distribution. So here you have a frequency distribution of the calcium content, and you see that the frequency distribution of the calcium content in the cement line is shifted to the right, so it's shifted towards higher mineral content. So this is fine. This is confirm what is already known. The cement line is more mineralized than the corresponding osteon. Then you find a little bit in between. Here is the frequency distribution of interstitial bone. Interstitial bone is in average older, so it makes sense that is also a little bit more mineralized than the osteon, which is then the layer inside the osteon are here, the one more to the left, and so less mineralized. Okay, this is fine. On the top of this, we did also more, let's say, local analysis, where we divided the osteon in several sectors, and in 12 sectors in this case, and we reported the mean mineral content in the cement line in each of these 12 sectors. This is to understand a little bit the spatial variation of the mineral content of the cement line when moving all around the osteon. And we did the same procedure for the different layers. So we want to understand if there is a spatial correlation between the mineral content of the cement line and the mineral content of region inside or outside the osteon. And I'll try to motivate this analysis later on when I present the result. So now I present the result of a single osteon. And so here what we see is that when we go from the layer inside the osteon, layer 3, layer 2, layer 1, to outside the osteon going through the cement line, we see an increase in mineral content because we said, okay, the osteon is more recently deposited, it's younger, so it has lower mineral content. The cement line is here, a little bit higher mineral content, and outside, the bone outside the osteon is older, is a little bit more mineralized. This is fine, is not so surprising. Cement line has higher mineral content. A bit more interesting maybe is when we look at the layer analysis, and here we were quite surprised to see that the mineral content of the cement line in each of these 12 sectors all around the osteon correlates very strongly with the mineral content outside the osteon. So this means that if you have a sector here where outside the osteon you have a higher mineral content, your cement line in this region will also have a higher mineral content. If you have another sector here which has a lower mineral content outside the osteon, the cement line which is formed in this region has also lower mineral content. So there seems to be a strong correlation between the mineral content of the cement line and of the external bone, even on, let's say, on different positions. And this is already one finding that we found very interesting. Now, what can we learn on the cement line mineralization by performing this layer analysis? Well, now I show to you some correlation, and I try to interpret this correlation with you. And while interpreting this different correlation, one thing to keep in mind is that we use the mean calcium content of the osteon as a proxy, once again, for its tissue age. This is not completely correct, but is the, let's say, in this context, is the best descriptor that we have for local tissue age. So what you see here now in this first plot is a correlation between the mean mineral content of the osteon and the mean mineral content of the corresponding cement line. They show a quite good correlation. This is already a result that was presented before when I discussed the previous literature. This correlation tells us that the mineral content of the cement line increases with time. So this is, the cement line is not, let's say, a static structure, but is a structure that mineralizes with time. This is fine. And from the fact that the mineral content of the cement line is always higher than the mineral content of the corresponding osteon, we can assume that the cement line has most likely a primary mineralization phase, which is faster than the one of the corresponding osteon. Because even for younger osteon, lowly mineralized younger osteon, the mineral content of the cement line is always higher than the one of the osteon. Then I correlate now here on the horizontal axis the mineral content of the osteon, the mean mineral content of the osteon, with the mean mineral content of the bone on a layer of bone outside the osteon. Well, what do we see here? We see only a very, let's say, weak or moderate correlation, and we can consider that the external bone outside the osteon, we can see that its mineral content is not much changing with time, so it's a kind of, let's say, horizontal, is comprised in an horizontal band, and this means we can assume that this, let's say, bone outside the osteon, interstitial bone or older osteon, are already in a secondary phase of mineralization, and so the mineral content increases very, very slowly with time. Most interesting is then when I correlate the mineral content of the bone outside the osteon with the mineral content of the cement line, and we see once again this very strong correlation. So this means that the mineralization of the cement line is not a kind of universal process, but depends locally on where the cement line is formed. If the cement line is formed, let's say, in a region with higher mineral content, where external bone has higher mineral content, also the cement line has higher mineral content. So these are a little bit the finding that we have here. I think I skipped this last one. So the take-home messages of this first analysis are the following. The mineralization of the cement line is different than the mineralization of the osteon. It has most likely a primary phase which is faster and where the cement line reaches a higher mineral content, and it has a secondary phase which is slower and, let's say, which is much slower than the mineralization of the osteon. I think most interesting is this other finding that the mineralization of the cement line is not universal, and this is what I try to sketch here. So this is, in the middle here is a young, newly deposited osteon, and the mineral content of the cement line of this newly deposited osteon depends on the mineral content of the interstitial bone, or let's say depends on the mineral content on the external bone. In a region where the external bone is more mineralized, the cement line is also more mineralized, and in a region where the external bone is less mineralized, as represented here, because maybe this is another osteon, also the cement line is less mineralized. And this may suggest actually some local recycling of mineral to build the cement line, meaning that the osteoclasts that are, let's say, excavating this cutting cone to build the secondary osteon, if they encounter a region which is with more mineral, they may also release more mineral, let's say, in this space, and this mineral is then captured and fixed in the cement line. And a similar process has been, let's say, discovered by Roschger and colleagues when comparing modeling versus remodeling sites. So also there they came up with the assumption of recycling of mineral in remodeling site, whereas modeling sites are more based on the transport of mineral. So yes, this is the first part. And now I like to discuss a second part of the work, which is more considering the mineral and mechanical properties of the cement line. And also here, I like to start by introducing some previous literature. Well, some previous literature seems to agree that the cement line appears to be a softer interface. So these works are essentially based on nano-indentation, where people, press a very small diamond tip in the bone, they measure the force and the displacement of this tip, and then they extract, let's say, local stiffness or local Young modulus of the tissue. So by performing several, let's say, indentation across osteon and interstitial bone, For example, Montalbano and colleagues reported that the cement line appears softer than the interstitial bone and then the corresponding osteon. And a similar finding were reported by Zuo and colleagues using AFM-based non-indentation is still similar to non-indentation, but using an AFM instrument. So I don't now enter in the details. Nevertheless, here, well, these are interesting findings, but we had a little bit some difficulties, let's say, to interpret this finding, considering that the cement line is more mineralized. So in general, a tissue which is more mineralized is also tends to be also stiffer. And one a little bit disadvantages of these techniques here, of these studies, is that, of course, they were very accurate in performing non-indentation, but then the difficulty is to locate the cement line in those results. Since the cement line is very thin, we believe we need to be, let's say, maybe we need a correlative approach to say, okay, I nano-indent in this location, but I need to be sure that what I'm measuring is really the cement line. So this is a little bit something that we wanted to understand a little bit better based on those initial findings. And then in terms of mineral properties, there is only a study which focused on the mineral properties of the cement line using tensor X-ray scattering tomography, and so tensor X-ray scattering, sorry, and reported that the cement lines tend to have shorter and thinner mineral particles. So this is a very interesting study, although is limited to a very small part of a single osteon. And we wanted to see if by analyzing larger region, by analyzing more osteons, we still retrieve those findings here. So our idea was to take the samples that we had previously characterized with for mineral content, once again, to look a little bit at regions where the cement line was clearly visible under backscattering electron imaging, to perform nano-indentation with a high resolution, high lateral spaces with a high lateral resolution. This is a typical picture of the surface that we have nano-indented, where you see each of these spots is the fingerprint of the nano-indenter, that we could visualize them with electron microscopy. The problem is that here we do not see the cement line, but we only see where we indented. So what we did here, we marked the position of the indenter. Then we used another approach, still based on electron microscopy, to visualize the cement line, which appeared in this region. And then we could superimpose the two images. And by superimposing these two images, we had the precise information of the location of the nano-indentation and of the location of the cement line. So we could classify the indenter, for example, falling in the osteon. This we call it inside, in here, on the cement line, here with orange, and on the interstitial bone. And we analyzed approximately 8 osteons, 9 regions per osteon, for a total of 5,000 indents. So, and then we classify this indent according to this different location. So we basically had maps looking like this, where each color represents, let's say, a value of mechanical properties. For example, tissue stiffness here. Let's consider this one, the stiffness or local Young modulus. And here is the corresponding local calcium content. And here is also a second harmonic generation imaging to see also the typical lamella arrangement of bone inside the osteon. Now, if we summarize a little bit the results, what we see here in this histogram, you see how the Young modulus changes when we go, let's say, when we try to approach the cement line. So the cement line is here. I still didn't plot the value of the cement line. But what you see here, you see a periodic variation in Young modulus corresponding to the lamellar modulation. This is something that is known in the osteon. So you have that, you have these lamellae, which are considered more, let's say, stiff and soft lamellae, depending on the orientation of the collagen fibers. And this is something which is reflected in this periodic modulation of mechanical properties. When I put also the cement line, so I collect all my points of the cement line, it appears quite clear that the cement line is not softer, but is actually stiffer than the corresponding osteon. So it has a young modulus which is about 15% higher than the corresponding osteon. So I will just, and also this is of course in line with the higher mineral content which has been previously also assessed, and this is in agreement with the literature. What is not now in agreement with previous literature is that here we found that the cement line has higher mineral content than the corresponding osteon. Interestingly, when we also plot the mineral content of we also plot the mechanical properties and the mineral content of the interstitial bone, we do not see that the cement line has, let's say, much differences in mechanical behavior with respect to the interstitial bone. Although it's less mineralized than interstitial bone, is not much stiffer than interstitial bone. So a little bit more insight on this relationship between composition and mechanical properties can be learned by plotting the Young modulus versus the calcium content. Since we have here information of each individual point, in each location that we indented, we have a measure of the stiffness or Young modulus and also of the calcium content, and we can correlate them, we can plot them on this plot. And what you see here is that we have all the points enclosed by this ellipsoid are the one of the osteon. The one enclosed by this more blue ellipsoid are the one of external bone, and here in orange are the one of the cement line. And what we see here essentially is that for the same, let's say, for having the same Young modulus, the cement line needs more calcium. So the cement line needs more mineral to have similar calcium, to have similar stiffness than bone. So this can be viewed also by binning the values of Young modulus in different regions, for example, from 22 to 23 gigapascal and so on, and seeing the required calcium content. And what you see here is that the cement line is always higher. So the cement line requires a higher amount of mineral to reach the same stiffness as surrounding bone. And we interpret this as a lower mechanical efficiency. How can we explain this? Well, we can explain this by looking at the properties of the mineral crystal. So we went to Grenoble, we analyzed once again the same samples by cutting small section with the microtome. We analyzed the same samples that we have previously characterized with QBI and nanoindentation, and we look at mineral properties, the properties of the mineral particles, like for example, the length and the thickness of the mineral crystals. And what we find, I think I skip a little bit here, we find that the mineral, that the crystals at the cement line are actually not thinner, but thicker than surrounding bone. This is something that we have obtained by analyzing several osteons and several regions, and they are also shorter. So the cement line has significantly different mineral crystals than bone. They are thicker and they are shorter. And we used a micromechanical model. I don't enter in the details. So we did some micromechanical modeling and we obtained the following result. For the same amount of mineral, a composite material which has thicker but shorter mineral particles is softer than the one which has longer and thinner mineral particles. So we have a micromechanical explanation on the behavior of the cement line, on the fact that the cement line is reinforced less effectively than lamellar bone. And we came up with this mechanical explanation. I think I can skip this because time is running out, and I would like to quickly discuss our last study on the interaction between the cement line and the lacuno-canalicular network. So in the last 5 minutes, I'd like to discuss about this. Once again, here we have some previous works which have reported essentially a disruption of the osteocyte lacuno-canalicular network when forming, let's say, a secondary osteon. In principle, what we see here are that the canaliculi of interstitial bone are cut off. So here it will be interstitial bone. The canaliculi here are cut off. And the canaliculi in secondary osteon, they tend to run, let's say, towards the cement line and then turn direction and runs along the cement line. with very minimal crossing of the cement line. On the other side, another work, subsequent work, which is based on SEM with acid etching, has reported that there are, in the region that the authors have analyzed here, they found many connections between neighboring osteons, and they also, across the cement line, and they also found that the canalicular crossing tends to decrease in edge tissue. So before presenting our own work, I'd like to introduce this recent paper of Anut and colleagues, which is based on the synchrotron radiation nanocomputed tomography. So they also analyzed this location of the cement line in a very different, let's say, very different bones, and they confirmed the presence of canalicular crossing. And also quite interesting, they observed these canalicular loops, especially in very young tissue. So this is an osteocyte, this is the cement line, and they observed that the osteocyte has canaliculi running towards the cement line, and if those canaliculi didn't find another osteocyte with another canaliculi, they turn back and they form this loop. And so this is a little bit two aspects which are very interesting. They've been investigated in a few locations in different samples. And basically, the authors here suggested that the canalicular connection across the cement line are not really random connection, but are a result of a systemic mechanism where the osteocyte is looking to connect to neighboring osteocyte located outside the osteon. Now, motivated a little bit by those findings, we wanted to explore also the interplay between the osteocyte lacuno-canalicular network and the cement line. We used rhodamine stain sample. We imaged those samples with confocal microscopy. We obtained images like this, where we see the osteon and the neighboring osteon and interstitial bone. Then we use TINA, which is a software developed by Max Planck Institute, colleagues at Max Planck Institute, to obtain a mathematical description of the network based on lacunae, edge, which are canaliculi, and node, which are branching point. So we have this mathematical description of the network, and a little bit our idea was to analyze especially the periphery, let's say, of the osteon. So we identify a 10 micrometer layer going from the cement line towards the inside aversion canal, and we characterize what is happening in this layer. So we look at we define as peripheral canaliculi the canaliculi which have at least one ending point in this layer or a starting point in this layer, and we classify the canaliculi as crossing, but not connecting to other lacunae, or as canaliculi as crossing the cement line and connecting to other lacunae. So this is a little bit the new analysis that we propose to distinguish, first of all, looking from all the canaliculi that are sitting in this external layer, in this, let's say, peripheral layer, how many are crossing the cement line and how many of these canaliculi are crossing and connecting. So this is now a little bit a picture which shows, let's say, the type of analysis that we perform. This is the osteon here on the left, this is the cement line, and this is the external bone. And we see that the majority of canaliculi are not crossing. Some of them are crossing but not connecting, and the one in green are crossing and connecting to lacunae, one from the osteon to the one from another osteon or interstitial bone. And here, I think the most interesting results are the following. We calculated by analyzing 14 different osteons from two individuals, we came up with 1% of connecting canaliculi among all the peripheral canaliculi, 1% are connecting lacunae, and this seems a small number, but thanks to this 1% of connecting canaliculi, 36% of the lacunae which are sitting at the periphery of the osteon, close to the cement line, is in a 10 micrometer layer, 36% of these lacunae are connected with lacunae outside the osteon. So this is a quite, let's say, a quite interesting finding, because it allows a lot of exchange, a lot of information between lacunae inside with lacunae outside. And we also found that around 7 canaliculi are connecting each pair of connected lacunae. So lacunae which are connected from the osteon to the interstitial bone or to other osteon are connected at least by 7 canaliculi. So these are sparse but robust connection. This will be my last slide, where we also or let's say my last result slide, where we also analyze, let's say, the orientation of the canaliculi in the osteon, especially when moving towards the cement line, and we distinguish the orientation as predominantly lateral or predominantly radial. And what we see is that canaliculi which are inside the osteon, away from the cement line, are oriented predominantly radially, whereas in the periphery, they are oriented predominantly laterally. So how do we put all of this together? Well, these are a little bit the summary of what we found so far on the lacuno-canalicular network. So the periphery of the osteon is densely populated by lacunae. The radial network in the osteon facilitates nutrient transport, let's say, from the aversion canal to the osteocyte lacunae located far away. The lateral network in the periphery is probably there to enhance cell-cell communication of all the lacunae which are sitting, let's say, close to the cement line. There is a decrease in network connectivity near the cement line due to the disruption of the network. So the network is indeed highly disrupted when there is the formation of secondary osteon. But nevertheless, despite this disruption, there is the presence of sparse but what appears to be functional connection, because they can provide structural pathways to connect to adjacent lacunae. And okay, this is a little bit a picture of the formation of this connection. When a new osteon is formed, some lacunae are most likely searching for, let's say, canaliculi located in the intracellular space. And this is somehow, let's say, is in agreement with the hypothesis of the previous study that I discussed. So I think this is then my last slide, where we analyze distinct feature of the cement line in terms of mineral properties, mineralization, mechanical properties, and let's say osteocyte lacuno-canalicular aspects. And I'd like to acknowledge my colleagues, of course, the students that performed this work, Astrid and Lena also, that work on the last part on the canalicular network, and our, let's say, founding partners, and all of you for the attention. Thank you.
Thanks a lot, Dr. Ruffoni. Now the session is open for questions. So you have two possibilities. Either you write your question in the chat, or you can also raise your hand, pushing the hand button in Zoom, and then unmute yourself. So we already have a question in the chat. So excellent presentation from Mohamed. I am so curious about connecting versus non-connecting canalicular. Does this reflect functional differences of related osteocytes? And does this reflect different subpopulation of osteocytes?
Yeah, that's a very good question, actually. We do not know. I think we do not know about this. The non-connecting canalicular is an interesting finding. Somehow, I would say the previous paper that I showed, that I discussed from the literature, showed that the canaliculi, or I assume that the canaliculi try to connect. If they cannot connect, they somehow form these loops. In our case, we also see that they just go through and they end up there. Why this is the case, we do not know. I think my simple answer is this: we still do not know. And we also do not know if this canaliculi at the end will close and the dendrite will retract. And right now we just see that there was a hole, there is a structure. We don't know if there is still the dendrite inside or not. It will be interesting maybe to understand if also those not connecting canaliculi are occupied by dendrites. I think this we do not know. That's a very good point, actually, yes.
Do you have any other questions? If not, I will ask one. I'm curious about the signal that the cells need to switch from creating the cement line and then starting to form bone and lamellar bone. Why do they start with the cement line and then go for the more lamellar bone?
It's also a very interesting question, which I'm not sure I have an answer, but as far as I understand, I mean, there is this first osteoclast at the tip of the cutting cone, they just remove. Then you have the secondary osteoclast, which are present, and this is the work of Lassen that I cited, which are present together with the pre-osteoblast, and they have a little bit the task of cleaning the surface and preparing the surface, I would say. And these pre-osteoblast, they deposit the osteopontin, as far as I understand, also from the work of Mark McNee. The other point is that what Lassen has shown is that there is a critical density of osteoblast which is needed, a critical threshold before the deposition can start. And I think in this time is where the cement line is formed. And the cement line, I think the basic understanding is that by the fact that it contains osteopontin, it also facilitates the adhesion between, let's say, the mineral and the collagen that is formed. One other thing that I want to say is that there is another work. I'm not sure if maybe I have it here. Let me see. One second. Well, there is another work that showed, well, it's fine, that showed that actually for a coordinate deposition of lamellar bone with long range interaction between the osteoblast, long range, I mean on a length scale much bigger than the dimension of the cells, you need a kind of ordered rigid substrate for this deposition to take place. And this could also be the role of the cement line to provide this substrate where then the formation of lamellar bone can take place. I think this is, but I think Natalie has maybe a question that is probably she can answer this much better than me, actually. Yes.
No, I can't. I'm just going to ask you another completely unanswerable question. I really enjoyed that, Davide. I keep thinking about how there has to be signals in the older canaliculi that the osteocytes in the new osteon are able to detect, but there's a cement line in the way and there's all of this mineral in the way. So, we as a field have to think about what are these dendritic processes in the newer osteocytes detecting that helps them know where to go to be able to connect with the previous canaliculi that are already there? Or do you think it's just they get lucky and that's why there's not so many of them crossing the cement line? How do you think that connection works?
Yeah, it's very good actually. We discussed this, right? Also, I don't know. I mean, they are very sparse, this connection, right? They are very sparse. So if there is really a consistent mechanism, I would expect to be a little bit more. On the other side, when I saw also the result of Anut and colleagues with these nice loops, these nice canalicular loops, then I thought, okay, this is probably not only stochastic, it's probably not only by chance that they can reach out. But how they can cross the cement line, I also do not know. If there are some locations where the cement line is not complete or things like this, and then they have the chances to reach out. Yes, that's. I don't know if looking maybe once again with the FIB-SEM, it's specifically those crossing and seeing a little bit around, especially maybe newly forming bone. I think this could maybe help if we can really do this, because the osteon that we analyzed are recently formed but are not under formation, let's say. But if we could target really newly forming tissue, I think this could help us to answer this. I would.
Yep, thank you.
Thank you. Nice to see you.
We have other questions in the chat. One from Dr. Ali Ghazemzadeh. So he says, great talk. What is the density of the cement line if you convert it to hydroxyapatite like VBMD in microcity?
That's a good point. I didn't do the calculation, but I think we can. There is with QBI, we have it in calcium content, and there is a small formula that you can convert this in a volume fraction, let's say. I would need to make the calculation. I do not know, but I can say that in principle is about 10-15% more mineralized than the corresponding osteon. So the amount of calcium is about 10-15% higher than the corresponding osteon. And I think this could give you a little bit an order of magnitude. Of course, with with micro CT, it's very tricky to get the cement line, right? The density of the cement line because of beam hardening effect. So you should then use maybe synchrotron radiation CT to get really numbers on the density of the cement line. I think this would be my suggestion.
Thank you. And another one from Marcin Kotlarz. Sorry for the spelling. Is it known if the thickness of the cement line differs at health and disease or as we age? Or if it changes as we age. I'm also interested in cement line at cartilage bone interface. Maybe you could shortly comment how this differs from cement line in the bone.
About the thickness of the cement line and the changes in health and disease, I do not know myself if this changes, and I'm not aware of studies showing that. And I would also not I don't think so. I would not know about a possible explanation for that, but I don't know. Let's say I do not know. Of course, since the cement line is a tissue that mineralizes, if you have mineralization, let's say, impairments of mineralization process in bone, it could also be that the cement line is impaired. So this, I would say, in disease, it could change. But I don't think people have investigated this much as far as I know, just focusing on the cement line. Of course, the osteochondral cement line is also something that we are very interested in. Now, well, I would say it has some features in common with the osteonal cement line. It is rich in osteopontin, is highly mineralized, and it seems also to be stiffer than adhesion tissue. In terms of formation, of course, it's formed in a little bit different ways, right? The way it's formed, because this has to do in the way, let's say, cartilage mineralizes essentially, and then this. Yes, so it probably is. Maybe the chondrocyte are then also involved in this, I think. So this could be a difference. But I saw that there was Michael Dowd online, and probably knows much more than me on the osteochondral cement line there. Yes.
Hi, Davide. Thanks
Hi.
for mentioning. I'm not sure it is high in osteopontin, actually. And I've been looking, when you mentioned Mike McKee, I started looking for a paper that showed that osteochondral cement line has high osteopontin. Calcified cartilage, the interterritorial calcified cartilage, does seem to be high in osteopontin already, which I only realized very recently. But I don't know any really good data, partly because I only just started thinking about it and looking for it. Anyway, that's an open question. I'm not sure it's formed the same way, and I think osteopontin might be sitting in the middle of it, but that's a new mystery. Anyway, thanks for the nice talk.
Thank you.
We have a last question. Well, if not, I'm going to thank you very much for the really great talk. We had a great attendance today, and we were really happy to have you. So thank you very much.
Likewise. Thank you very much for inviting me, and all the best. Bye-bye.
Bye.
Bye-bye.